Welcome everybody back to our afternoon session. We're glad to have you with us, and we're honored this afternoon to have our first presenter be Dr. Paul Kruszka. He is the Chief Medical Officer at GeneDX, and he leads the genomic research on rare diseases and promoting access to genomic medicine. So welcome, Dr. Kruszka. Thank you for being with us this afternoon.
Thanks for having me. I appreciate it. I'm going to go ahead and start my slides and get going here. Okay, let's go to slideshow. Okay, so, well, first of all, I just want to thank the Hope organization for inviting me to talk today. This is certainly an honor and privilege to talk to this group. I don't think I've been with the Hope group since before COVID. It's been a long time since I've spoke with Hope. So I worked at the NIH for a long time as a staff clinician, and have attended, I think, most Hope conferences since probably 2012. So for over 10 years, I've been affiliated with Hope. Starting out with Scottish Rite when Hope was held there, and of course it was held at the initial one was at NIH, and then I think the last one I went to was at the Great Wolf Lodge in Mason, Ohio. That must have been 2019 or somewhere around there. So the introduction, I am a physician, clinical geneticist. I really specialize in genetic testing. And when I was at the NIH at my first Hope conference, was great. We had a lot of people, their families and individuals affected with holoprosencephaly. I asked a lot of parents what they thought we should be researching or doing at the NIH. With Max Monk, I worked with Max Monk, who really started a lot of the genetic research, and overwhelmingly the answer was, can you find the reason why my child has holoprosencephaly? For a number of reasons. Can we have more kids? Was it that the few beers I had when I was pregnant and I didn't know I was pregnant? Was it the M&M candy that I ate a lot of when I was first pregnant? All these reasons, they really wanted to know why, and often it is a genetic cause. So I wanted to talk about genetic testing because that's really my specialty today. I do work at a company, so that's a disclosure. GeneDX, we're a genetic testing company. I'm chief medical officer. I retired from the government 3 years ago, and we diagnose a lot of holoprosencephaly, genetic causes of holoprosencephaly at this company that I work at GeneDX, and there's many other companies like that. So certainly highly recommended that you get the appropriate genetic testing, because there's a lot of implications based on the type of result you would get back. And I can tell you, the company I work for, and you'd have to check with other companies, but certainly if you have Medicaid insurance, there's zero out of pocket at our company. If Medicaid doesn't support genetic testing in your state, we eat the cost. So I think that's one of the reasons I work at this company, but very important. There really should be no excuses not to get the testing that you deserve and want. So with that, I'm going to talk a little bit about genetic testing, and we'll have time at the end for questions. I tried to design this talk so folks can ask questions if there are any. So I'll do a quick introduction. I'll talk about genetic testing mechanisms, the different types of genetic variants, exomes versus genomes. This is a very important, probably the most important bullet I want to talk to. The genes that cause holoprosencephaly, and then what's coming in the next 5 years in genomic technology as far as diagnosis. As everyone knows here, rare disease is rare in itself, such as holoprosencephaly might be rare, but combined, it's common. So rare is common in the US. There's over 7,000 rare diseases in the United States, affecting 25 to 30 million Americans living with rare disease. 80% of rare diseases have a genetic cause. Most are pediatric, and are pediatric onset. And the average time someone goes from symptom onset to diagnosis in holoprosencephaly, that would be birth to diagnosis, or maybe even ultrasound, is about 5 years. So it takes a long time to diagnose holoprosencephaly or rare disease in general. There's definitely a time lag there, unfortunately, and I want to talk about that today. One thing that comes up a lot and that we have to be careful about with holoprosencephaly and all rare disease is this term actionable. And insurance companies will use actionable, doctors will use actionable. For example, why should I do genetic testing on holoprosencephaly if there's nothing we can do with that information? So this term actionable, I just want to spend just a few minutes on. As everyone here on this call knows, that when you get the diagnosis, you can connect with other families. Certainly in the case of holoprosencephaly, you don't need a genetic diagnosis to connect with families, because you know from the ultrasound or MRI that holoprosencephaly exists. So you can connect with other families. And I think I first learned with Hope that the power of connecting with families and how important that really was to share information, not to feel isolated. So Hope has taught me some great lessons going forward in my career. Number one lesson is really always listen to families first. The end of a diagnostic odyssey is I believe, actionable. Families often cross state lines, travel, and seek multiple tests and specialists to get a diagnosis. Once you know the diagnosis too, you have more information on the history and prognosis of the disease. And although I don't know of a lot of precision therapies yet for holoprosencephaly, there will be some, I'm sure, in the future. I want to point out that there is a shortage of medical geneticists in the United States. Sometimes it's hard to get a genetic test from a non-genetics professional. I'm certainly trying to change that. I think everyone should be comfortable ordering genetic testing if a child has holoprosencephaly, or even an adult. And as you can see, each one of these circles, the smallest circle is 1 FTE, that's full-time employee. And there's large areas of the country that there are no clinical geneticists. Often I've wondered if there's more astronauts than geneticists in this country. I'm not sure about the answer to that. So let's move on to genetic testing. Genetic testing has progressed. I know there's some families in Hope that have family members that are now adults and have seen the full evolution of genetic testing. And I'm going to tell you, it's changed and will continue changing. This field is under constant change, and the timeline goes back really hundreds of years, starting with Gregor Mendel, who first figured out the laws of inheritance. He was an Austrian monk who did some experiments with some pea plants and figured out hereditary and inheritance and how this all works. In the 1950s, Down syndrome was described by a physician named Lejeune, Dr. Lejeune. This is having 3 chromosome 21s, and certainly this type of research has benefited holoprosencephaly because some common causes that you can diagnose with a karyotype, which I'll go over a little later, is trisomy 13 and trisomy 18. So that's the 1950s. In the 1970s, a sequencing named Sanger sequencing was discovered, and this was, you were able to look at our genetic code, all the letters of our genetic code. And this is important because when I first started working with Dr. Max Munka, we were using Sanger sequencing in his laboratory when I first joined. The human genome draft was was reported in 2001, the first draft. And this really allowed us to know how many genes do we have and what's the reference genome, which I'll talk about in a little bit. And then next in 2004, next generation sequencing technology started. And I'm going to give you a really good example of this. This is the state of the art now. When I joined Max's lab in 2011, I really pushed for Max to really go ahead and start applying next generation sequencing of holoprosencephaly, which we did thanks to you, thanks to all the samples that Hope provided. We learned a lot, and I'll touch on that a little bit during this talk. One thing I want to say is that in 2021, the American College of Medical Genetics put out this guideline for genetic testing in general. And what this said was that if you have a congenital anomaly, you should get an exome and genome, and that should be first-tier testing. I'll come back to this concept over and over during this talk. If you have holoprosencephaly, I'm going to repeat it as many times. You should be getting exome or genome sequencing, not panel, single gene, or any other type of testing. And this is supported by the American College of Medical Genetics. And most insurance payers have picked this up. So if you have United Healthcare or Blue Cross Blue Shield, Aetna, any of these insurance companies, they will pay for this type of testing. And like I said, if you have Medicaid testing, at least in our company, we will not turn you away, even if we absorb the cost. There will be no out of your pocket if you have Medicaid insurance. So very important. I want to break down barriers and myths in this talk about testing. So state-of-the-art sequencing is something called short-read sequencing or exome sequencing, genome sequencing. And basically, you guys will send in your samples. Either you'll swab your cheeks or a blood sample. We do the sequencing in our laboratory. And really, I can generalize this to any university or laboratory does this. We did the same thing in NIH. And then once we do the sequencing, we do this data analysis or bioinformatics where we use computer methods to figure out what's going on. What is the genetic reason why you have holoprosencephaly? And I'm going to try to make I use this with medical students, so I'm going to try it out on you guys. This is a cartoon. And what we're going to do here is we're going to assume that the biological father has blue DNA and the mother has red. And notice that this is the code, your AGCTs. Now, normally you have 3 billion base pairs in your genomic code, but just to make it simple, I made it just less than 100 here, just to make this a cartoon and simple. But remember, in real life, you've got 3 billion of these AGCTs. So you send us in your sample from your child first. And remember that your child gets 1 chromosome or half their DNA from the biological father and half of the DNA from the mother. So that's the mother is red and the father is blue. And certainly the colors don't mean anything to me. We can switch them, but for this cartoon, I made, like the sheets in the in the hospital. The blue ones are for boys and the pink ones are for girls. So just to keep it easy, that's how I picked these colors. So you send us the sample, right? And we get a sample. The first thing we do is we break up the DNA into little pieces. So we got bits and pieces from mom and bits and pieces from dad. And we do this either by a chemical reaction or we use ultrasound energy and we break up your DNA. And when I say we, this would go for any commercial lab or the NIH when I was there or a university. This is how we all do it, how we use this next generation sequencing or exome genome sequencing. And remember in 2001, we figured out the reference genome. What we do is we use computer methods to line up all these bits and pieces to the reference genome and see how you compare to it. And so we just keep on taking these pieces and aligning it to the reference genome. And this is how it looks in real life. We get these big printouts like this. And we try to figure out for the reference genome, in this case, the reference genome has a G and mom has on the maternal chromosome, there was an A. And so we try to figure out what's going on here. This is not matching the reference genome. So this is the really simplified way of what we do. And we look for all kinds of different genetic variation based on this. The first one, this is called a karyotype, where we've got 23 sets of genomes, or we have 46 chromosomes. And in this case, the child has Down syndrome because there's an extra chromosome 21 trisomy. This could have been chromosome 18 or 13, which are causes of holoprosencephaly. We can also pick large chunks of DNA that's missing, called copy number variants. And I know there's people at Hope that have these copy number variants, because I've seen a lot of your reports over the last decade or so. And this is a deletion or duplication that's more than 1,000 letters. A nucleotide's a letter, one of those AGCTs. We can also pick out a smaller deletion, like just like one letter gone. So we're using a sentence here as an example. See the dog run. And if you remove one of these E's, a small deletion, you get set, head, org, un. Or if you put an extra E in there, you get CF, Edo, grew. So these are examples of how just adding one letter to your genetic code can make things go haywire with these sentences. And then also you can have just one change in your letter. So you have an A to C here, and this changes the amino acid from histine to proline, and just shows you just one small change can really change your health. All right. I'm going to pause for questions at the end of this, so just hold your questions. So I know there might be questions on this. Now, I'm going to keep on repeating this. Exomes and genomes really are the way to go. And by the end of this presentation, you'll understand why I'm saying this. So a genome has 3 billion base pairs. That's our genetic coding. And an exome has 1% of that, or 30 million base pairs. And the exome just covers the protein coding region of the genome. This is where most mutations lie. So exome traditionally has just been a more economical way of looking at your whole genome. And I want to tell you the difference between an exome and genome now. And I'm going to put it in the context of the Gettysburg Address. So the Gettysburg Address is 7 years ago, our forefathers brought forth. So let's say that the Gettysburg Address is what we call your exome. Okay, it's just the protein coding, or really the important part. The in-between letters, when you take this as a whole, is a genome. We used to call this junk DNA, this WPOD, $AMFKU. It's just gibberish, and it's always meant gibberish until recently. Now we're learning that it is important and that it's worthwhile doing a genome if you could get away with it. Again, a lot of the reasons we started out doing exome because it was smaller and cheaper. A genome in 2001 would cost $92 million. Now the cost of doing a genome is dropping drastically. When I made these slides, 700, now it's probably about $500. So it's dropping drastically. When I was in Max's lab, when I first started there, a commercial exome was costing patients or insurance companies $20,000. So ridiculous amounts of money. That's all changed. And here's a graph. Moore's law is a graph of computing power, and you can see that genomes have quickly dropped off from Moore's law, the cost of computing power. Now on this graph in 2022, it shows it at $1,000. I can tell you now it's dropped to $500. Again, insurance companies are covering exome and genomes now. Universal coverage, I would say most insurance companies cover exomes, and we're seeing more and more insurance companies starting to cover genome. For example, Texas Medicaid will cover genomes starting in September. So really, the payers have come around in a big way to do this. Also, if you think about it, a lot of infants with holoprosencephaly will end up in the NICU, and there's a lot of payer policies to that are supporting rapid genome sequencing in the NICU. So certainly if you have a child with holoprosencephaly, they could get an answer in a few days from this thing called rapid whole genome sequencing, which is really fantastic. And we do a lot of that at the company I work at right now. So I want to talk about genes that cause holoprosencephaly. And I was looking for a picture of Max Munka, and this is the one I found on my first one that came up on my iPhone. Max and I, we went to India together in 2017 to give some talks about holoprosencephaly and other rare diseases. And these are a few of our Indian colleagues. We're at Manipal University here, but this is a recent picture of the most recent picture I had on my iPhone. Max left the NIH a few years before I did. He left in 2019 to become the chief executive officer of the American College of Medical Genetics, and he did that for 3 years. Now he's completely retired from the medical enterprise, and I still keep in contact with him. I haven't seen him in about 1 year. It's the last time I had breakfast with him, but he looks just like this still. He hasn't changed much. All the way to the left is a picture of me during this trip. I guess it's been 7 years ago for sure. And our two of our Indian colleagues, and this man's name is Garisha. He is our closest colleague, and this is one of Garisha's colleagues here at Manipal University. And here's a paper that really is the first genetic cause of looking at a gene. And this was a paper by Max Munka when he was at the Children's Hospital of Philadelphia, and Eric Rosler. I worked with Eric as well at the NIH, and this is from 1996. So we're talking now about almost 30 years ago. And Max really did all the groundwork in early genetic causes and came up with this gene called sonic hedgehog, which I know a lot of people have heard of here. Max did not pick the name sonic hedgehog. It was already a known gene. It had already been named. Max just associated sonic hedgehog gene with holoprosencephaly. So he had nothing to do with the naming. It's kind of a bizarre name for a gene. But some wacky scientists came up really with this name, and Max just figured out it was in holoprosencephaly. Max decided to study holoprosencephaly serendipitously when he was a fellow training at the Children's Hospital of Philadelphia in the early 90s. His first patient that he was called to go see was an infant with holoprosencephaly, and he realized that there wasn't really much known about the genetic causes, so he started working on it. So that's really how all this came to be. So this is 1996, 30 years. And traditionally, these 4 genes were associated with holoprosencephaly: sonic hedgehog, ZIC2, SIX3, TGIF. And this was before 2016, before Max and I really started doing a lot of exome sequencing, next generation sequencing, to discover what's the other causes, because we were only getting an answer about 20% of the time on cases we analyzed, and we really wanted to bump that up. So we started doing exome sequencing on everyone and trying to draw some conclusions from that. And to the right are all genes that we added to this list. STAG2, which is X-linked, as is SMC1A. So we showed that holoprosencephaly can be X-linked as well as autosomal dominant, which was the usual cause. RAD21, SMC3, FGFR1, PPR112A, and CNOT1. These were all genes that we were adding to this. And I know you're going to get a talk by Seth Berger. He's still trying to add more genes. I believe there's a lot more genes associated with holoprosencephaly still than this model. I can tell you all of these genes on the screen, we diagnose a significant amount of infants with this at my company, GeneDX. I was going through it last night, and these are all genes that we now diagnose, and thanks to the work that Max started at the NIH, these genes are official causes of holoprosencephaly. However, I think there's a lot more causes we've left. There's a lot still on the table. There's still families not getting the diagnosis, and I know I'm 100% convinced that there's a lot more causes that we just haven't gotten around to yet. So there's still a lot more work to be done in the arena of finding a cause. Again, you really need to do the exome or genome test. Here, this is the company I work for, so I'm going to beat up on this company first. You can see none of the new genes are on our panel that we would do for you. So I don't see STAG2, SMC1A, CNOT1, SMC3. Let me see, is FGFR1 on here? Blah, da, da. No, I don't see FGFR1. So not even FGFR1. And some of these genes on here are not strongly associated with holoprosencephaly. So here's an example of a panel and that you really better serve by doing an exome or genome. And this is the company I currently work for. And you're probably asking, why would a company that a holoprosencephaly researcher works at not have this? It's very difficult to upgrade panels. We had hundreds of panels. We're discontinuing most of them. We'll eventually discontinue this one too, because it's too hard to upgrade. Every time you upgrade a panel, you have to go back and get CLIA CAP certification. You have to validate it. And really, the small companies that do genetic testing don't have the bandwidth to update these panels. And now I'm going to pick on some other panels. Here's Invitae, another company that was recently acquired by LabCorp, and you can see they just have 1, 2, 3, 4, 5, 6 genes. So really not having a lot of different genes. And here's another company. So I want to put other companies up. These are all common companies. Prevention Genetics, very good, very, very good commercial company. And you can see that they're missing a lot of the genes that we discovered, because all companies have this issue. They've got hundreds and hundreds of panels. They can't keep up. So really the only way we're going to find new genes is do exome and genome, because collectively we can start collecting data and publishing these and making sure that the word is out that there's new genes associated with. So again, this is a recurring theme of my talk that I've that I continue to go over, and that's why why are we doing panels still, and what are the disadvantages of this? So this is a I actually did this cartoon because I didn't want to have to mess with trademarks and everything. One thing is this comparison to this person right here. This purple person has lost their key. So this is a common analogy used in genetics. And this person's only looking under the lamplight for the keys, even though this is not where the person lost the keys. And a police officer in the story walks by and says, where'd you lose your keys? And this person says, I think over in that parking lot across the street. And the police officer says, well, why are you only looking here under this lamp and not there? And the person says, well, this is the only place I can see. This is all I can see. And genetic testing is very similar to this. We only look where we can see. So a panel, for example, only looks at the genes on the panel. We only look where we can see. Various types of technology only looks where they can see. And so if it's not under the light, we are missing it. So that's why I think there's still more work and there's new technology coming. I told you about the short read sequencing. I gave a cartoon, an example of that. That's really the commercial state-of-the-art genetic testing right now. I will tell you that there's something called long read sequencing. So state of the art, as I mentioned, short read, long read. There's some new technology that's very new, unproven, that I'm looking at in our company, and I know Seth Berger is looking at too, who will talk to you later. He was actually one of our fellows, one of Max's fellows at the NIH, as I was. He's a little younger than myself, but I passed. When I went to the commercial industry side, I wanted to pass the torch to someone to keep on doing sequencing. So long read sequencing is the next piece of sequencing. And on this cartoon right here, what we have is we have a gene A. This could be any gene, and these bigger rectangles are the exons or the exome, the protein coding, and the little thin line is the supposed junk DNA that I had talked about. That's part of the whole genome sequencing. And the short read sequencing, remember I told you we break the DNA into these little pieces, where these are the little pieces that I was talking about. Long read, so these pieces are 120 base pairs long, so 120 pieces of our alphabet, and we align to the reference genome. Now, long read sequencing, these pieces are 15,000 base pairs long. So they cover a lot of these genes. And they fill in the gaps, if you can imagine. These short reads leave a lot of gaps. And down here are three reasons or gaps they fill. One is structural variation. That's where the structure of your DNA is different. These things called repeat expansion, where you have repeat alphabet or genetic code. Now, this is important too, because I can tell you in the gene ZIC2, one of the variants or mutations we find in holoprosencephaly is actually a repeat expansion. So this is important. Phasing, that's just telling where the gene came from, or mom or dad. That's why we do trios for short read sequencing, so we can do this phasing. You don't need to do phasing with the parents with long read, because you can kind of see the whole gene. And then there's something called pseudogenes. There's a lot of genes that we have that have these pseudogenes, genes that are 99% or more similar. And short read sequencing gets confused because we don't know how to align the short read. So there's a lot of different reasons out there to do long read sequencing. And like I said, this is emerging technology. Hopefully it will be commercially available, cheaper, and really the idea is to find more answers for you guys. So really important. Steve, does this go to 2:30 or 1:30? Is that right? I think so. I think this goes to 1:30. So I'm going to try to wrap it up as soon as I can here, because I want to give time for some questions. Here's my email. Please, getting a little feedback. Please feel. I think someone needs to mute.
Yes, you do go to 145, Paul. I think
Okay.
you're muted.
Okay.
Did you hear that?
Okay, I go to 145.
Yeah.
If you guys could just mute for a second, I think we're getting feedback. Okay, so here's my email, and I want everyone to be, to feel free to reach out. And if you have any questions about holoprosencephaly, I'm happy to answer them. But I wanted to make this a discussion as well. It looks like we have more time than I thought. I originally thought this was 45 minutes. So what I'm going to do is open it up to questions, and I might have to bring my slides back based on that. There's a lot here in previous HOPE meetings. There's been a lot of questions about genetic testing, so I'll try to answer them the best I can. So if you have if there's any questions out there, I'd be happy to entertain them. While we're waiting for questions, again, the largest take-home message really that I wanted to bring home to HOPE is if if you have new members that are looking for a diagnosis, okay, someone, Rachel asked, how do we ask questions? One is type them in the chat. But what I wanted to say why these questions are starting to come online is that if you have new families that are coming into Hope, or your family in particular has had a panel or no genetic testing, please start with an exome or genome, because you're really going to be missing. You're going to be missing a lot with the panels. Like I said, the panels are becoming obsolete. And I'm not putting a plug in for our lab. Certainly, any university that you're going to, any laboratory, there are many places you can get exomes and genomes. And as I tried to show, that insurance companies cover this. So, and if an insurance doesn't cover it, please email me. And certainly I'm want to get you guys answers. So that's important. So, okay, I'm going to start with questions. It says, are gene variations the most common cause of holoprosencephaly? I'm not sure, to tell you the truth. I think genetic causes are the most common cause of holoprosencephaly for sure. What we know is the non-genetic cause, the most common cause, is type 1 diabetes. We've shown that in a few studies that really high sugar levels can be a factor associated with holoprosencephaly. So when we say gene variations, single gene variations, my sense is it's probably about equal with trisomy 13. Trisomy 13 is a very common cause of holoprosencephaly. That is a genetic cause. That's where you have an extra chromosome 13. I can say that the gene variations that cause holoprosencephaly usually have a better survival. We see older children and adults with gene variations where there's very few infants that survive longer than a few years with trisomy 13. So across the board, I will say that a genetic cause, certainly the most common cause, but single genes, which I think Eric is asking, it's probably a tie with that, with trisomy 13 as a cause. So Eric, please type in more questions if I didn't sufficiently answer that. Okay, the next question is from Jody. It says, my daughter is adopted and there's no contact with her birth family. Is there any advantage to genetic testing for her? Absolutely, yes. So yes, definitely. I think there is an advantage. It's good to know the reason, because different genes have different prognosis, different conditions associated with it. Obviously, the reproductive counseling won't be able to take place with the biological parents. And also, if your daughter's diagnosed, it will add to the general community of knowledge. So it's better to test. If you're testing both parents and the child, because that's called a trio. However, we don't need a trio. It's not absolutely necessary. Just by doing genetic testing on the child, we'll still be able to pick up a lot of genetic variation. There's this concept called de novo variations. That's when the child has the mutation and the parents don't. And that's the most common cause, or most common scenario, I'm going to say. However, there's a lot of times where variants are inherited. We see that even with sonic hedgehog, where parents will be, from what we can see, not affected at all. From what we can see. And they'll have a sonic hedgehog variant, or they'll be minimally affected and they'll pass it on to their offspring. And the child will be more severely affected. We don't know why that is. And we call that penetrance or reduced penetrance or variable expressivity. We don't know why that is in not only holoprosencephaly, but a lot of other genetic conditions where people are looking for these reasons. So it certainly can be inherited. And so that I think this is a great question because it brings up the concept of de novo inheritance, again, where the child has the mutation, the parents don't. However, in holoprosencephaly, we've seen so many times where we go back and the parent does have a variant. So, but if it is de novo, that's a stronger argument, as you can imagine, that that's the variation of interest. So I will say that, you know, hopefully, you know, I'm Steve, Leslie, I'm certainly open to coming back to holoprosencephaly next year, or hope next year or the year after. And a lot of these questions, it just reminds me of memories were sometimes answered over dinner, breakfast, Bobby, or even over a beer at the Wolf Trap Lodge. You know, a lot of these questions, you know, were in-depth discussions. And you know, during COVID, we were doing a lot of these types of things. And as we're getting back after COVID, certainly invite me back, because I can talk forever about at least about genetic testing. That's mainly my expertise. Is a laboratory based physician. So I definitely keep abreast of holoprosencephaly, and we you know, at GeneDX and other commercial labs, we have a lot of data, and I really want to we want to share that data, and we need more people doing research in holoprosencephaly, at least the causes for sure, because there I believe there's a lot of data, and there's a lot more answers out there that we haven't uncovered. We've only uncovering the tip of the iceberg, starting with the work that Max did, and then later I joined him for the last few years of his career at the NIH. Okay, so I'm going to go to the next question, and this is by, hey Jessica, I know the Miller family. Almost had my career shortened. I remember we were about 10 years ago, we were over at Scottish Rite Hospital, and they put me in the back of their van, and we took a turn so quick, and we had all kinds of stuff, including their daughter and me moving around. But I love the Millers, and Jessica, it's so good. I wish we were in person so I could give you a big hug. But yes, I was hitchhiking, and you know, they were the family that pulled over and picked me up and threw me in the back of their van. So Jessica, her question is this: how many genes now are being looked at, and how many are being confirmed? Here's my point. I don't think we should be looking at a fixed number of genes. That's why I'm strongly suggesting, and I'm backed up by the American College of Medical Genetics, that we look at all 20,000 genes and everything in between all the genes, all 3 billion base pairs with a genome or exome, preferably genome. I think the days of panels are gone. I don't know if you saw this, Jessica, but I had a slide where I showed some major companies, including my company where I work, but other commercial companies that are just missing this. They're looking, you know, anywhere between 4 and you know, maybe 50 genes, and that's unacceptable. We need to look at all 20,000 genes, because the panels are not covering the newly discovered genes, and we're not really gonna find any new genes if we don't look broader. So certainly, we need to keep on looking for answers and advancing. Okay, so Margaret is the next question. I know Margaret too from years ago. I know Margaret's from my home state as well, from Michigan. And Margaret, the Barrio Mike family I've known for a long time as well. And her question is, what do the exome and genome results tell us? Will we understand the results? The genetic defect could be a new one in the individual, and non-inherited, correct? That is important to know for siblings. Yes, that again, that's the point about reproductive counseling from this for the siblings. Do the siblings have it? Is the siblings affected health going to affect their health? So we're talking about the health of the siblings, but then also when the siblings get old enough to start having children of their own, that question will be remained. And I have a good story that kind of illustrates this example. When I was at the NIH, I was called by a young woman, and she was getting ready to get married. She lived in Chicago, and he said that 3 of her brothers had died of with holoprosencephaly as a result of complications of holoprosencephaly. And she wanted to know if she would have children with holoprosencephaly. Since she had 3 brothers, it was obviously inherited. And I got her information. I talked to Max, and we figured out that her samples were frozen in Max's lab. And so I reanalyzed those samples of the brothers, and the 3 brothers all had a deletion, a big deletion that when I was talking about on those slides, those copy number variants encompassing ZIC2, the gene ZIC2. And so I realized that the dad also had it. He was unaffected from, I'd never met the father, but from what I was told, he was pretty much unaffected. So I told this woman, I said, listen, we know why your brothers have this. I want you to go to your doctor who initially saw these your brothers, and she was still in practice in Chicago, still practicing at Lurie Children's. So I called the doctor up, and we got her a test, and she had not inherited the same copy number as her brothers. So this is an example of, yes, it's good to know what it's, knowledge is power. It's good to know what's going on. And it also illustrates that things change over time, that just because you had a negative result in the past doesn't mean you'll have one in the future. Technology changes, our knowledge of the genetic code changes. So this is a family that didn't have an answer when they were seeing Max in the 90s at Children's Hospital of Philadelphia, but certainly by the time they recontacted us recontacted Max at NIH, there was a change. So that's the example I have. Let me see if I answer this question fully, though. It says, what do the exome and genome tell us? Okay, so they tell us, the changes, and they can tell us a lot of changes, from a single alphabet change, single letter change, to a copy number variant change. And yes, Margaret is demonstrating this concept that I talked about, the de novo situation, where only the child has the variants and the parents don't. So it was a change in the germline, the sperm or the egg was the genetic change, and not in the parents. So so we call this de novo, only the child has, and that's the usual scenario. But we always want to test the parents, because we do have scenarios like the one I just outlined, where a parent will pass it down, and we don't know why that is. Again, it's a mystery to the medical community, and we're still looking into that. Is there a modifier gene? Is there something else going on? Why, in the case that I just told you, the Chicago family, did the father have the mutation ZIC2 and was unaffected and had 3 children with holoprosencephaly? We don't know that answer yet. Or why there's families on this call where I know that the parents are not as severely affected as the children. All right, I'm going to go to the next one, J&A Hilliard. It says, my daughter had genetic testing when she was first born about 4 years ago. I'll have to dig up the results. But will it be clear and obvious on the results if she had exome or genome rather than a panel? If she already had a panel, what justification can I present to her to get retested? will that help her simply add? So I really hope that somewhere there should be that it was a panel or exome or genome on this. Certainly you know, ping your doctor. They should be able to tell you what test that was done for sure. If it was a panel, it's going to be missing things. So definitely there's justification to have exome or genome. And the justification is, I'll just share my screen for 1 second here. There's a couple of justifications. The first one is this. Panels don't, okay. Panels, let me see here. Panels only cover a limited amount of genes. We have 20,000 genes. And the genes that Max and I worked on before he retired was STAG2, SMC1, A, SMC3, CNAT1, PPR112, CNAT1. None of these genes are on the panel that I work at the company. The other company, that's a big company in Vitae, none in that panel, and none at Prevention Genomics. So there's one justification for you, and I'm happy to talk to you know, any doctor that needs justification. The other justification is that it is the standard of care now to do exome or genome for congenital anomalies such as Holoprosencephaly. If you look in most payer guidelines, you look at the standard of care. The standard of care is to do exome or genome. So this is not experimental. We have tons of evidence and justification to do this. So, and certainly email me, you know, if you need any help, that you should be having an exome or genome. So good question. Next one is from Rachel. And the question is, my daughter recently had a genetic test from Kennedy Krieger. I honestly don't remember the results, but I don't recall them saying it could be passed on. She was also tested, wow, 2001, that's a long time ago, when she was born, and we were told she had nothing genetic. She has Centellencephaly and is very disabled, but very healthy. Follow-up, is KKI a reliable place? So Kennedy Krieger, I believe, is a reliable place. We actually had a fellow in Max Munka's lab go there named Mahim Jain, who practices there. So it sounds like in this case there is an answer. So the question is, what is that answer? You should know that answer for sure. You should know if it was positive or negative. You should know the results. And you should know whether this can be passed on or not. So you should know all this information with this test. So you need to schedule another appointment and go through this, because you have to know what is the gene or the variation, and how was it inherited? Was it de novo, so it's only in your daughter, or it sounds like you guys don't have it, the parents don't have it. So this is all incredibly important, and it's unacceptable that they didn't tell you or tattoo it into your brain or your soul, because these are things you need to know about your daughter, for sure. And also the type of testing that was done. And like I said, Kennedy Krieger is generally, they're affiliated with Johns Hopkins. They're generally a pretty decent place that I trust. Kennedy Krieger. One thing about Kennedy Krieger is they also do a lot more with therapy, interventions, and treatment than we did. Max and I really focused on the biology and the testing and the answer. We were not really as good as Kennedy Krieger. We used to have a physician from Kennedy Krieger come to the Hope meetings for a while. I think he's left Kennedy Krieger. His name escapes me. But certainly Kennedy Krieger has always been a good place for treatment as well. So yes, Rachel, please find out all the questions, because those answers should be clearly on the report. And I would be curious, Rachel, to know how everything works out with you. And I would also be interested to know what on the report you don't understand. And as someone who makes reports, how can we make reports better readable for you? So I would like to know, just in general, for what can we do as a ecosystem or community to make reports more readable for everyone, because everyone needs to understand this information about what's going on with your child from these reports. And just not to digress too much, but I was at the last Hope conference that was in it was down in Dallas at Scottish Rite. It must have been some numbers of years ago. I was walking out to the parking lot, and or out to the little drive around to catch a ride, probably from Jessica. And a woman stopped me on my way out there, and she says, "Listen, I don't know why my kid, my son, I remember his cute blonde-haired kid, still remember, I don't remember the name, but I remember the child, about a 2 or 3 year old child with blonde hair." And the mom says, "I don't know why my child has holoprosencephaly. Can you just look at his report?" And so she pulled out this folded commercial report. I forgot which laboratory. I opened it up, and the child had a deletion, a big deletion encompassing ZIC2. So this was obviously the reason why this child had holoprosencephaly. But that bothered me, that the report wasn't clear enough to this mom that we had this, random passing by. And that encounter left an impression on me, like, what do we have to do better as an ecosystem to make this crystal clear? Why does your child have holoprosencephaly? Okay. Yes, the next question is from Jaziel. And the question, I hope I said your name right. But the question is, are there specific symptoms or change of prognosis related to specific genes? And yes, there's a lot of changes how you're affected. The most drastic one was there's a condition, there's a gene called, it's the rarest of all the genes, called CNAT1, and you have to check them for diabetes. That this is sugar diabetes, not diabetes insipidus, that often goes along with holoprosencephaly. There's definitely different prognoses. ZIC2 often has different facial manifestations with clefting, not having clefting, things like that. There's every gene, excuse me, some genes have congenital heart disease associated with them. So every gene is very different. And has a different effect on the child. So definitely, it's good to know the gene. Not all genes are the same. Okay, so okay. My husband and I were also tested. This is Rachel again. Nothing they brought up, but still, we need to know everything. Okay, the doctor that was at Kennedy Krieger. This is from Leslie Hartley, who I've known for a long time. I met her at my first holoprosencephaly meeting, which 2011 or 2012, whatever. I forgot their odd or even years, but a long time ago, I met Leslie, who was in leadership for Hope, and she says Eric Levy. I'm almost positive he's not practicing anymore. Okay.
He's not.
I think he's moved on, but still Kennedy Krieger is good with managing all the other things that goes along with holoprosencephaly. There's other things that can go on, like diabetes insipidus and other things. So I'm at the end of my time, but I just want to you can email me if you have more questions. I'm happy to answer them. I put my email on a slide, and then the Hope Foundation can grab it. And then there's also Seth Berger that will be talking to you. He's also still working on discoveries, telling me he needs to write his most recent paper so we can all learn more from him. But and just I think in closing, I just want to leave you with one thought. A panel is not sufficient anymore for diagnosing holoprosencephaly.
Can you hear me, Paul?
Yeah, I sure can.
Okay. Well, it's Steve. Leslie's still around here and doing things, but me being the president now, but I know one of the things I want to do is invite you to a podcast at some point here in the near future, because Susan and I do podcasts. And I think that would be really good to have you on there, and maybe even more than one, where we can kind of talk a little bit more and share some information, and definitely want to pull you back into the Families for Hope family. So
Yeah,
we miss
yeah.
you. I think everybody else.
I would love to do that, and I still think there's a much more work to be done.
There is.
From the discovery piece, the part that I'm involved in, the learning more about answers and the biology. So I think there's a lot more to do. And I think there will be treatments, different treatments going forward for, we're starting to see things that, that are also have birth defects and brain, changes, like there's a, Angelman syndrome. There's a lot of things happening out there. So certainly would love to be involved. Okay, so yeah, please invite me back. Stay connected.
We definitely will.
And if you have scientists that need information or collaboration, let me know, because I am working with a few people. There's Rob Lipinski and other people that are still looking on some causes of this, and yeah, there's a lot more to do for sure.
Oh yeah, we'll definitely
Thank you.
bring in mind. Appreciate you coming on today and talking to everybody, and everybody has some great questions. I think they were really, really good questions, and I know I've got a bunch, so I'm going to save them for the podcast.
Yeah, we'll be looking forward to talking with you again soon. Thank you so very much.
Yeah, you know where to find me. Thank you.
Yep.
Okay.
All right. Have a good day.
Thank you. So we're going to be joined by our next presenters. We're going to connect them to us here. They're going to be talking about family caregiver support and what that is and why it's so important to family wellness. We're going to be joined by Angela Shierk, JD Torre.
There she is.
Hello, welcome you two. Well, we're a little bit late starting. I'm just going to let you guys jump in and introduce yourselves. We're so happy you're here, and thank you for joining us. So we'll get started.
Well, I'll start. My name is JD. I am an occupational therapist. I'm kind of a fresh one. I just graduated in May. I had the opportunity to meet everybody from Families for Hope through my last semester of school, which is when we did what's called our capstone. And essentially it's kind of like our big research. Well, mine ended up being research at the end of our program, and just got the opportunity to meet everyone there and work with them, and just kind of fell in love with the whole organization. And then my capstone ended up turning into something that carried over through after school was done. So that's kind of how I ended up here and stumbling into here. So I'm just very excited to be here with you guys, and hopefully introduce something that can be really helpful and impactful for you guys. So that's a little intro about me.
Thank you.
Thank you, JD. I'm Angela Shierk. I'm an occupational therapist and clinical researcher at Scottish Rite for Children. And I have the opportunity to be JD's capstone mentor here at Scottish Rite. So I will give him all the credit for the presentation that you're about to hear. And I'm really just a support and a backup. So JD, I will let you take it away.
So essentially what the purpose of today is to introduce something that we've put together called Parents Supporting Parents. And if ideally we would kind of like this to be a little bit interactive, because the point of this group is to really hopefully launch what we are calling a support group. I even heard you guys talking at the beginning just about how, I've never met another person with HB, and it'd be nice to meet people, and obviously in person is always ideal, but since we do live in a little bit more of a virtual age, there's other ways that we can help with that at least a little bit, whether we meet through Teams or FaceTime or Zoom or whatever it may be, and that at least helps some of that feeling. So in my capstone project, what I did my research on, so I always feel a little bit weird talking about this experience because it's so minor compared to what um, all of you are going through. But I was first exposed to just what it's like to have to be a parent and a caregiver with my own son. My firstborn was born about a month early. That was a very hectic day. His his mom went in just for a regular checkup, and 30 minutes later, I'm driving to work, and she's like, "We're having a baby today. We didn't have a crib. We had a high chair, which he wasn't going to need for about a year and a half. I don't even know if we had a car seat, to be honest. Like we were just not prepared. We thought we had more time, and he ended up being in the NICU. He was tiny. He was, well, my version of tiny. Again, everybody has different scenarios, but he was about 4 pounds, 7 ounces when he was born. He was smaller than a football. I was a first time parent. I've been around kids, but now I have this like itty bitty tiny thing, tinier than most babies. And he's in the NICU and he's got tubes attached to him and everything. And we were fortunate to only be there for a little over a week, but I met a lot of families that were there who had been there for months. I met a family that had been there for over a year. And just being able to talk to some of the other parents and just seeing what they were going through, having to juggle both being a full-time parent and also a full-time caregiver, even having that feeling while they were in the hospital. So that was the first time I was kind of minorly exposed to it. And then when I went through school, during my clinical rotations, I was able to really see it in real life because I actually worked in home health. So I got to see it outside of the hospital setting. It was actually in the homes, working with families who had children that had all sorts of different diagnoses. And the thing that, even though all of the situations were a little bit different as far as like what the child was specifically going through, that feeling of how this impacted the parent's life on a day-to-day basis, that part was the same. And my heart was just really, really heavy for it. And we would just have open conversations about just how they would feel and all of that. So a lot of the research that I found about what parents go through when they are having to be in this role of both a full-time parent and a full-time caregiver, none of it's going to be surprising for any of you because you're living it every day. But the one that stood out to me the most was that sense of feeling isolated. There was a lot of quotes from different parents that were talking about how they've lost a lot of their social group and social support, and not even necessarily because people just like intentionally abandoned them, but it's if you're not going through something like this, it's really hard to know how to support someone that is. You know, you may think you're saying the right things and you accidentally say the wrong things, and then it ends up hurting your feelings or like whatever it may be, just because they don't know. And then over time, it just fizzles out, and then you just feel like it's just you. So the purpose of these support groups that we're wanting to initiate is to help with that sense of feeling alone and to create a safe space for parents of children with HPE to meet other people that have similar experiences, gain the social support, and just really help create an environment to just be able to connect with other people that also know exactly what you're going through. Because even us as people in the healthcare and medical world, we still don't even really know the emotional impact that you're going through on a day-to-day basis. So it's even different trying to talk to them because they're coming from that medical lens and it's very black and white. It's completely more impactful to be able to talk to other people and they can look at you and say, I completely get what you're saying because I've been through XYZ, versus a doctor that's just like, I understand. Like they're trying to be empathetic, but it's just not the same. So that's what the whole purpose of these groups are. And again, for today's purpose is to just give a little preview of what that would be like. Like that's what we're going to be doing in the breakout rooms. We also would love to learn from any previous experience that any of you may have with support groups, whether it is through here with Families for Hope, or if it's somewhere outside, just what kind of experience you've had with support groups and what's worked, what's maybe not worked. What can we improve on? Because we have had support groups in the past, and some of them have done really well. Some of them have started off strong and kind of fizzled out over time, which happens. But we want to know how can we keep this going to where it's still effective for everybody that's involved, and then hopefully just gain some connection with other people that are just like you. That's really the main purpose of today. In the support groups, it's always best that everybody's just kind of on the same page as far as what the expectations are, what the guidelines are. And the thing that's most important with any support group is that it is a judgment-free zone. No emotions are wrong. I even go as far to personally say that no emotions are bad, because if you're feeling it, there's a reason that you're feeling it. It's more about what we do with the feeling. But I don't think there's such a thing as bad emotions. People having an equal opportunity to share or not share with because you can get a lot just by sitting back and just hearing other people's stories. And if you're not ready to share, that's okay. But that everybody has the opportunity to share if they want to. And when they do share it, that it stays within the group. Having that trust with each other that what I say is safe and it stays here, that's really, really important in any group. And obviously, which I don't think this is going to be a problem with any groups that are done with Families from Hope, but just being courteous and respectful of each other. So just all the basic things like don't interrupt and blah, blah, blah. All that stuff we know. And okay, so let's prepare for the breakout room. So what we're going to do today, because we're going to partner you guys up in either groups of 2s or 3s. And what we're going to do is not necessarily like interview each other, but is find 3 things that are common between the 2 of you, but it cannot be related to anything medical. It can't be related to HPE or anything like that. We're looking for things like, I don't know, like favorite colors or like hometowns or, favorite places that we visit or like places on your bucket list. We want things that are outside. We talk about HPE a lot, like, and every day. That's literally what our life is consumed by. So we're going to take a break from that and find some commonalities between the 2, or like I said, 2 or 3 of you guys. And the goal is 3. That's the goal. So like I said, we will break out into those groups. We'll give you guys, we'll probably give you guys like 15 to 20 minutes, somewhere around there. That's probably plenty of time. And just let you guys connect and find those commonalities, and then we'll come back together. And like I said, those that are willing to share, we'd love to find out what you find out about each other and the commonalities that y'all have, and then also just talk a little bit more about like the vision for the support groups that we're hoping to launch. So
while we're doing breakouts, Susan or Steve, do you want to give an overview of kind of the history of the support groups that have been in place in the past and kind of what that has looked like and which ones are still going, maybe?
So a lot of it started, well, it honestly goes all the way back to when they had the Yahoo groups. And so there were different groups on there. And there's some that still exist on Facebook, but it was, kids that have trachs or kids that have seizures, or, with Families for Hope, we had a small group. And so as things have progressed and, Facebook and social media, most of what we have now is we have a group for our bereaved families. And then we have a dad's group. That's just made up of all, different dads. We have the main group that's the Families for Hope, but it's the private Facebook group, and we have the public one. But the private one is meant to be for moms and dads, so that they can, be able to share information with each other. I'm almost thinking we're going to have to start one soon that's just the extended families, because a lot of times I'll have families that will invite aunts and uncles or grandparents or something like that onto the private group. And sometimes I have to kind of weigh the good and the bad, as far as sometimes maybe families want to keep things just a little bit more private with what they're discussing and things. And so I try to keep that main group to where it's just the moms and dads, because, you know, we have a different, there's a different way that you want to communicate with each other maybe than have your extended family always know what's going on and things. And so we have that. So it's kind of, those are what 3-4 groups I think that we have now. And I know that we have some of our families that probably talk to each other as a group outside of our groups. So I know those kind of exist as well. So that's why when we talked about this, you know, even what we started last year, kind of talking about what this would mean for our families, and that, you know, we could have a whole new group that's geared towards, you know, the caregivers and the mental health and everything else. So,
a bunch of people.
so I'm excited about the opportunity to do that.
It
But
just says they
so
don't
those are the
have
four,
access.
pretty much the 4 groups. Still trying to figure out how to split us off into the rooms. I
Well,
think.
we're split into the rooms, but some people, it says they can't have access to the breakout rooms.
We have some people in the rooms.
Yes, we do.
So some people could get into the breakout rooms and some can't. So they're not breaking out.
If you were in the breakout rooms, just kind of give us like a quick summary, or just some things that you guys found that were in common for those of y'all that were in the breakout rooms. While you guys are doing that, while we were hanging out in here, essentially just what the purpose of us doing that is just to show that even if all the details aren't necessarily the same or anything like that, like just having an environment where you can just talk and just connect to people and just help create that sense of connection is really the whole point of the support groups. The support groups are supposed to be there to obviously like share any kind of emotions that you're going through or up and downs that you're going through and your experiences, and just be able to talk to people that are going through something very, very similar to you. But it's also just be able to, you know, just kind of have people that you can just step away from for a little bit, and, you know, just talk about other things in life and just create that connection and know that you are not going through this journey alone. And that's really the whole point of the support groups. If answers come in on the chat, you guys just interrupt me because the chat doesn't update for me. So I'm not.
I think Angela put on ours the commonalities we had were we all want to go on vacation. we all like comfort food. We like a variety of music. And then we all drove old cars with a high number of people who can drive a stick shift.
They do, which is actually pretty impressive.
Yeah.
The dying aren't these days. So for what we would like to hear from you guys, and again, if y'all can maybe put in the chat, has anybody here ever been part of a support group before? That can be like I'm sorry, I might be moving too fast if y'all are still typing from the breakout rooms, but has anybody been in any support group before, whether it's even if it's not necessarily related to HP or caregiving or anything like that? Does anybody have any experience with that?
Jessica Millar says yes. And then I can say yes for myself.
I'd say yes.
Angela, yes.
Eric, no.
No. Just a Facebook support group, that still counts.
I mean, that's still something, yeah.
Another one, yes, just starting up. A lot of yeses.
Okay, good. So for those of you guys that have been a part of a support group, if you could put in the chat, what are things that made you want to continue to be part of the support group? I know that might be kind of a little bit to type, but what were just things that made you either just want to join one in the first place, and then, or what made you keep going back to it if you did? I know for me, I was part of a support group, and I think at first I honestly didn't really, it was virtual like this. And at first I didn't even necessarily know why I was wanting to come back. I just did. Something just kept pulling me to go. And The first several times I really didn't speak a whole lot, which if you know me, that's surprising. But but I think it was just being able to even hear how other people have gotten through a similar experience that I had gone through.
Yeah.
That's what kept me. It was more the hope that some people were able to give that were on the same journey as me, but maybe like 200 yards ahead of me. So just it kind of let me get out of the right now, if that makes sense.
Well, I think we got some folks on here. Connection, community, joint understanding, building connections and a sense of community. And then some, Rachel put, desperately trying to find anyone who is in the same situation as us. My daughter is so rare and virtually nobody I know understands. And Rachel, you're in New Zealand, right? Is that, or am I getting mixed up? Knowing I wasn't alone, knowing a safe place.
He's in Baltimore.
Oh, Baltimore, okay. So you don't have anybody in Baltimore. Well, we'll try to see if we can't connect you up with some folks. Mental distraction from my own life to hear others' perspectives. So, well, I think with support groups I've been in, it was, well, like one of them was with Families for Hope, and it was just the dads.
Mm.
You know, but I think the other thing too is with that is you're trying to, you know, the dads are maneuvering, you know, having a child with HPE and just everything else. And so that was one of the ones. So I think the ones I've been in have always been like, if there's a specific reason or a specific, I don't want to say gender, but that seems to be, because it was mostly guys, it seems like, but.
It was like a specific niche almost.
Yeah, because I remember years and years ago, there's a book called The 5 Love Languages, and people always talk about that still. And so I led a group, and it was all guys, but you know, lo and behold, within that is I had like 2 or 3 guys that were either contemplating divorce or were in divorce. And as a result of our group, they reversed, you know, they didn't get divorced and stayed married and stuff. So it's like a lot of the groups that I've been in or done, that's a specific thing.
Yeah. Okay, next question for you guys. What would you see as potentially being the biggest barrier for you being involved in a support group, whether it's through Families for Hope or just in general? What is potentially the biggest barrier? Because our goal is to be able to help overcome those barriers so you can get what you need. So we'd like to hear from you.
Yeah.
So far, everybody's answering time. I get that.
Yeah.
Yep. That's, what was the question again, Rachel? Oh, what would your biggest barrier be in joining a group or being in a group? Jessica, yeah, Jessica is in a unique situation as she's the sibling and not the parent because both of her parents passed away in the last year.
Wow.
So she's in a different, and we do have some families that that's been the case.
So mainly time is the big one. So
it sounds like, yeah, time.
Okay. So a realistic, and just kind of asking everybody here for realistic goals, do you think that once a month is attainable? Like, is once a month something that we could probably work out? I think once a week would be too much. Or is once a month not enough? I don't know.
Let's see, I got a yes.
Okay.
Bi-weekly to monthly, yes. Once or twice a month is reasonable. No barriers. Texting is not done.
Okay. No.
Not fun. Texting is not fun. Yeah, I agree. Sometimes the textings can be a bit much.
So I see the preference would be to do a group where we're actually together like this and
Yeah.
talking.
Yeah, no, I agree. And that's what they would be. They would be something where everybody could have their cameras and mics on and be able to share and all of that. Ideally, we would. So here's a food for thought, and this is kind of what we would be doing tomorrow. So tomorrow's group or meeting on the caregiver support is mainly about people that would be interested in potentially being what's, I guess, what we would call like a facilitator, just someone that's in the group that kind of helps guide and steer the group itself, because we want this to be led by other parents. We don't want this to be led by people. That's the whole point of this group, is parents connecting to parents. But we do need to have someone that's kind of not necessarily like in charge of everything, but like, they're the ones that has like, they're the host of the meeting on Zoom, or, they just kind of help like guide the conversations or whatever it may be. So it would be completely voluntary, and it's just important for us that it's led by people that can also reciprocate that shared experience part. Because even if like I was leading a group or anything, that kind of defeats the purpose of what we want the group to go, just because I'm not living what you guys are living through. So we would like to invite anybody that would potentially be interested. Coming tomorrow does not mean you're a facilitator. That's not what it is. It's just getting information on what facilitating a group would look like, and we'll also do a little bit of training for that, and what like some common obstacles like you would run into in groups or anything like that. So just, it's a kind of more of a general info. This is what we would need for people to lead that. So if you are potentially interested in that, please come tomorrow. That's what we're going to be doing today. So it'll be slightly a little bit more educational than today was, because it'll be more about what being a facilitator actually looks like. But that is a big thing for us, is we want it to be led by other, not even necessarily just parents, because as it was just pointed out earlier, caregiving isn't always just that one role. It could be any role. So whatever role you fit into the family, if you're kind of in that caregiving position, that's what we would like to be kind of the head of our groups. Outside of that, that's really all that I have for today. Thanks for being patient with me as I worked out the kinks and everything on my part, but I appreciate you guys being involved and sharing in the chats and answering questions and all of that. And it's just a very big honor for me to be here with you guys, and I appreciate you even letting me be in this space for a little bit. So that's all that I have.
We're thankful to you for all the work that you've done, and excited for you to be back tomorrow to talk with people who would be interested in learning more and perhaps considering facilitating. And I want to thank everybody else for all of the thoughts and sharing here in this space. And so, Angela, JD, thanks so very much, and looking forward to having you back tomorrow.
Us too.
Okay. For being with us.